about here. Did you see that slide?
I think it was identified.
This one?
I think.
So you've seen this slide for sure.
Uh-huh.
Have you seen this one?
Yes.
What about this one?
Oh.
I think this is where I stopped. What?
Yeah, that's where you stop, because that's where I stopped looking at.
Okay. Well, let's see if I can get rid of some quizzes. The average on this quiz was actually quite good, 8.2, and both sections were right at 8.2, so y'all are, well done. Michael? Lorna? You guys go to play volleyball tomorrow, right? John? Anybody? Larry? Katie? Allie? Hannah? Chase? Carly? Lizzie? Lindy? India? Little girl, not doing nothing, but your name is spelled way too. Isabella? Harmony? Grace? Anthony? Working out good. Connor? Well, that was fun. Let's do it again. All right, put away like before, whenever you're finished, go ahead and turn your paper over if you would. And this time, follow my instructions and make me a picture. Be wrapping it up in the next couple of minutes. Okay, go ahead and fill in the last blanks and pass them over this way, face down, please. Reading your truth. today's my killer day. I go from now until like 7:30 at night without stop. What does a bagel cost at Einstein's? Huh? Okay, good. I've got like $4. Cause I don't get lunch today. I gotta have put something in my stuff. Okay, let's see here. A solution with pH of 4 is acidic. In phospholipids, the blank is hydrophobic, fatty acid tail. The blank contains the portion of DNA which forms ribosomes. Nucleolus, yeah. Fats and oils are composed of 3 blanks bonded to a glycerol, fatty acids. Testosterone and cholesterol are classified as lipids. The DNA of a prokaryotic cell is known as the nucleoid. Let's see here. DNA combined with protein forms chromatin. Cellulose is a polymer of, it is a carbohydrate, but it's a polymer of specifically glucose. There we go. Specialized rotary structure of bacteria, flagellum, and we'll see. No one turned me down, did they? Okay. All right, so quizzes. Let's see. The consensus seems to be that this is the next slide in our repertoire, if I can get it to play this one. So here's what we'll pick up. What this is, I think we did touch on this the other day in class, but I wanted to mention that this is a picture of the nucleus. Sometimes people think we're looking at a cell with the nucleus here, but the whole thing is the nucleus of the cell, and you can see this is the nucleolus, which is where what gets made? Ribosome. Well, it contains the information to make ribosomes, yeah. Okay, so the chromatin in the middle here is a mixture of DNA and protein primarily. And then I'll point out that the nuclear envelope has numerous pores in it, and the membrane that makes up the nuclear envelope is perfectly normal phospholipid bilayers. It's exactly like what we see on the surface of the cell membrane. This stuff here is the beginning of the endoplasmic reticulum, and you can see all these little dots attached to it. Those are ribosomes. In eukaryotic cells, you have some ribosomes that are actually attached to the endoplasmic reticulum, and others that are referred to as free ribosomes, okay? In both cases, they make protein, so don't confuse that. And I'll reiterate that the nuclear pores are basically the way that stuff gets into the nucleus as needed or out of the nucleus as needed, but not, it's very tightly controlled, secure area, so to speak. If it were the door in the administration building, it might say, do not enter, unless you have a key or whatever, staff only. So, endoplasmic reticulum is, a lot of people, when you first start studying a cell, you know it's got a nucleus, you know it's got some organelles, and if you position all that stuff in the cell, it still seems like a big empty sac with these things tucked away in here and there. Honestly, every spare bit of space in a cell that's not an organelle is like, if you can imagine saran wrap or plastic wrap or something like that, just stuffed in there as tight as you can get it. That would be kind of how the endoplasmic reticulum would look like in a cell. It's extensive pathways. You can see how closely tucked together they are here. These little bumps on the endoplasmic reticulum are the ribosomes I was talking about. And here's a diagram showing the rough endoplasmic reticulum, which is where, as I said, where protein gets made by the ribosomes, and then the protein gets channeled off to other places, particularly the Golgi apparatus, which we'll get to in just a minute. Okay, and then the smooth endoplasmic reticulum here is more involved in lipid synthesis, lipid manifestations, if you will. It's particularly, the smooth ER is particularly abundant in cells that are involved in detoxifying things. So if you look in the liver, for instance, where a lot of your detoxification of poisons and products, alcohol, whatever, that get broken down, you'll find a lot of smooth ER in those cells more than, say, in a muscle cell or something of that nature. Ribosomes come in large and small subunits. We'll talk a little bit about that later on. Interestingly, it's possible to distinguish eukaryotic ribosomes from prokaryotic ribosomes, and we'll be getting to that in a few minutes and talking about the significance of that. It's a pretty darn important research that was done in that regard. So here's a free ribosome. They may or may not attach themselves to the endoplasmic reticulum. If they do, there's a receptor here which allows them to kind of dock like a boat at the yacht club. And when they start making their protein, the chain will get secreted into the channel in the ER, and then it'll move off through the endoplasmic reticulum in the direction of the Golgi apparatus. Speaking of which, here it is. The Golgi apparatus always reminds me of like a stack of saucers in your china cabinet or something, you know, it's a continuation of the endoplasmic reticulum, but it's special. It's not where protein gets made, it's where protein gets finished in a lot of respects. Once a RNA molecule has been translated into protein, that is usually not the final product, okay? In most cases, those proteins have to be folded into special shapes often. They may have to be edited somewhat, some bits or pieces removed, so there's a lot of what we call post-translational. What would that word mean? Post means after, and after translation, okay? So after the peptide has been made, then there's typically some post-translational modification before you actually get the final protein. Mostly that happens in the Golgi apparatus. And the other thing the Golgi apparatus does is it packages those proteins and ships them off to where they need to go. You can almost think of the Golgi apparatus as being like, well, you all probably been to Rouse's at some point in your 2 weeks here by now, right, across the street. We've got a grocery store over there. Anyway, the manager of the grocery store, and there's Rouse's all across the Gulf Coast, there's lots of them. And it's a big store, it's got lots of things. So the manager there realizes he's starting to run low on, what's he running low on? Steaks, okay? He's running low on steak. He's running low on dish detergent, and he's running low on toilet paper, okay? So what he does is he sends a message, by computer probably, not probably for sure, to central shipping and receiving, wherever that may be. They'll package that kind of stuff up on a big semi-truck, and it'll come here to Mobile, and they'll unload the steaks, the dish detergent, and the toilet paper at the Rouse's, okay? That's basically what the Golgi apparatus does, but with various types of proteins and potentially other chemicals as well, but mainly proteins. And where do those packages go? Well, some of them, so here's the endoplasmic reticulum. The proteins are made here. They pass through the endoplasmic reticulum, and they form up in little vesicles. So these are little bubbles, little vesicles full of protein. And they come into contact with this side of the Golgi apparatus. Now look at it. This is named cis, and this is named trans. You're going to encounter those terms repeatedly in biology and in chemistry, actually. Cis means on the same side. Trans means over on the other side. Okay, later on we'll talk about cis elements and trans elements when we start talking about how genes get expressed. When you take organic chemistry, Dr. Borders is going to talk about cis configurations and trans configurations. So the list kind of gets, and there's other things, it gets longer and longer. So at any rate, cis means on the same side, okay, so the same side as endoplasmic reticulum. Trans means the other side. And as these vesicles move along the Golgi apparatus, they actually kind of shuttle along the edge. It's called vesicle shuttling, okay? But as they do that, any kind of post-translational modifications that need to be made, get made. And eventually, and they also get labeled with an address. So, Rouse's, Oak Hill Road, Mobile, Alabama, or whatever, okay? And the address tells them where to go. Some of those vesicles may stay within the cell. Some of them may contain proteins that are useful in the mitochondria, for instance, in which case they'll go over, and because of the address that they have tagged to them, they'll attach the mitochondria and spill their goodies onto the inside. Some of them, if it's a secretory cell, like a salivary gland or something like that, some of them are going to go over and fuse with the cell membrane and open up and spill the contents to the outside, maybe digestive enzymes or something of that nature, okay? You all with me? Okay, so that's the Golgi apparatus. Golgi apparatus is an underappreciated organelle in the cell. It's got a lot of work to do, just like Rouse's shipping and receiving department. Other things, so here comes a primary lysosome. What's a lysosome? Anytime you see that suffix "-some" inside a cell, you're talking about some kind of organelle, typically a vesicle of some sort. What does that word, that prefix "lys" suggest to you? You know what lysis is. Lysis? No? Okay, put this one in your biology toolbox. L-Y-S, L-Y-S-I-S, word, derivations like that. They mean to break apart, to tear down, to digest, something like that. In prokaryotic cells, they have digestive enzymes, for instance, that are being secreted into the cytoplasm, but they're in such slow concentration that it's not going to affect the cell. Eukaryotic cells have much more powerful enzymes in much greater quantity, and if they were released out into the cytoplasm, they start digesting the goodies in the cytoplasm of the cell. That would be a bad thing. That's actually what happens pretty much immediately when you die. Just in case you're wondering what happens cellularly after you die. Your lysosomes start breaking open inside the cytoplasm of the cell, and you get digested from the inside out. Sorry to bring that irritation. Anyway, so what happens is, here's a lysosome. It's full of digestive enzymes, but it's sequestered away from the cytoplasm, so it won't do any damage to the cell. It's going to come over here. Here's a phagosome. There'll be other vesicles that contain things that need to be digested. In this case, this cell has phagocytized, absorbed, if you will, some kind of nutrients from the outside, and it's contained in this phagosome. What's going to happen is this phagosome is going to come over and fuse with the lysosome, the primary lysosome. Once that happens, this is sometimes referred to not that cleverly as a phagolysosome, but in any case, digestion takes place inside there. Any kind of nutrients that need to be absorbed will be diffused out of the vesicle, and then what's left would be waste material, okay, and it would be expelled to the outside. So this was a grossly oversimplified statement of what goes on with vesicles and lysosomes and so forth inside the cell, but it's a good introductory view of it. What do we have here? The mitochondrion. You can tell because number one, it's a double membrane structure, so there's an outer membrane and an inner membrane, and that inner membrane has all these little cristae inside it. In fact, this is very diagrammatic. A real mitochondria would have 10, 50 times as many of these folds on the inside. Again, in biology, what are folds about? Surface area, totally surface area. If the inner membrane was just kind of clung to the outer membrane, then the enzymes that associate themselves with the inner membrane would not have as much area to establish themselves, right? It's the same with other structures. Your intestines have villi and various things called crypts of Lieberkühn that are folds inward and outward and so forth. If you could take your intestinal mucosa and stretch it out so it's a flat sheet, it would probably come close to covering the soccer field, okay? I mean, it makes up that much different. You're aware, perhaps, that your brain has a lot more folds in it than, say, a fish brain or something like that. What's that about? Where are the neurons? They're just underneath the surface of the gray matter, right? And so, not surprisingly, because we require more neurons to carry out our chemistry classes and biology classes and things like that, then thank goodness for those folds, right? So it's all about surface area. The enzymes and cytochromes, we'll talk about this in the second block of material, that are associated with aerobic respiration, the primary ones, are attached to these cristae on the inside of the mitochondrion, and boy, do they do their job. The chloroplasts in plant cells, or other photosynthetic cells, are somewhat similar to the mitochondria in that they do have an inner and an outer membrane, but they not only have an inner membrane and an outer membrane, they have kind of an inner membrane called the thylakoid membrane. One of the few structures that you'll find in membrane systems inside a prokaryotic cell, bacteria, is in a group called the cyanobacteria. They're the ones that invented photosynthesis. And so they have little thylakoid membranes inside the cytoplasm of their cell. But for the most part, they're pretty much the only significant group of bacteria that actually have an internal membrane system of any kind. Once again, you can see the inner membrane and the outer membrane are kind of smooth, but check out the thylakoid membrane. It's folded, but in a very different way than the cristae of the mitochondria, isn't it? Again, it looks like little stacks of plates or something like that, but by having all these stacks and folds, what does that do? It increases the surface area of the thylakoid membrane where all the enzymes that are associated with photosynthesis are stored. The more of those enzymes, the more efficient the photosynthesis will be. So, important point. Someone, I remember running through this section or the other section, asked me the other day about whether mitochondria, I think, or chloroplasts had their own DNA. Well, in fact, they do have their own DNA. It's a single circular chromosome, just like other bacteria. They do have their own ribosomes. They can make some of their own proteins. And remember I mentioned a few minutes ago that you can distinguish eukaryotic from prokaryotic ribosomes? Did I mention the name of a woman named Dr. Lynn Margulis the last class? Okay, so Lynn Margulis was a professor. She's still alive, but she's retired by now. She's getting to be a pretty elderly woman. She was at the University of Massachusetts at Amherst. And when she got started back in the 60s, people were wondering, okay, what the heck? Explain the existence of mitochondria. Explain the existence from an evolutionary perspective of chloroplasts, for instance. Where do they come from? Why do we find some very chloroplast-like things in bacteria and some very similar things with similar biochemical pathways in certain eukaryotes, plants, okay? And Dr. Margulis, new faculty member, trying to make a name for herself, let me just tell you, Dr. Margulis is one of the most brilliant people on the planet, all right? I've met her once. She's also one of the most obnoxious people on the planet. But I kind of understand it because she was coming up with some really profound ideas about where mitochondria and chloroplasts came from. And everybody thought she was a dumb, newly minted assistant professor at this university. And shut up, woman, you don't know what you're talking about. We're the big guns. We know better than you do. Well, she was coming up with the right ideas, okay? And so she got mad about it. And she basically said, what she was saying was that mitochondria and chloroplasts are in fact bacteria, endosymbiotic bacteria. What does that word mean, endosymbiotic? Symbiotic means living in or with, right? Endo means inside the cell. So she said bacteria are mitochondria and chloroplasts are endosymbiotic bacteria living inside eukaryotic cells. They originated way back in the days for mitochondria, probably before photosynthesis was invented, okay, when the very earliest of the eukaryotic cells were around. And then she said about everybody goes, you're an idiot, go to the back of the room. And so she started testing it, and she says, okay, if they're prokaryotic cells, what kind of DNA do they have? Well, they have a single circular chromosome. And you know what? They have a single circular chromosome. If they're prokaryotic cells, bacterial, endosymbiotic bacteria, then they would have prokaryotic ribosomes. So she checked it out, and you know what? They had prokaryotic ribosomes. And the list, there's some other things. Those are the two strongest pieces of evidence. There's a variety of other things that she brought forward as well. So by the time we made it into the mid-70s, everybody's going, boy, I feel really bad about being so down on Lynn Margulis, oh my God. But she was ripping mad by that point in time, and she was publishing books, and she was taking some pretty hard shots at those people that were trying to shoot her down earlier on. Rightly so, right? She won the contest. When I was working on my doctorate at the University of New Hampshire, she came and gave a presentation on her research with videos of where she thought cilia and flagella originated. I mean, really convincing stuff. And it was a phenomenal thing. I've seen two superstars in my life. I mean, hardcore superstars. She's one of them. And I also saw one of Janis Joplin's last concerts. And in both cases, man, they just entered the room and they reached out and grabbed every person in that room and just jerked them to the front of their desks or their seats and held them there for an hour. Okay, so sorry you guys won't get to see their motivation. Yeah. So anyway, it's called the endosymbiosis theory, and it's what everybody understands the deal is with bacteria. Okay, so there's a lot of other organelles in eukaryotic cells that are surrounded by membranes. Peroxisomes, gloxosomes. Peroxisomes, again, bacteria, peroxides in general are toxic to cells, okay? There are peroxides that bacteria have to deal with. They have some enzymes to deal with them, and they don't have to deal with them in large quantity. But, I mean, what did your mom do when you got a boo-boo on your knee? She overwhelmed the bacterial systems by doing what? Put peroxide on there, right? Okay? And so eukaryotes have much greater need of various peroxides, and so they have these, again, just like the lysosomes, they have these peroxide-containing vesicles called peroxisomes to carry out whatever needs they have for the peroxides. Gloxosomes tend to be things that are involved with carbohydrate and or lipid synthesis as well, so they sequester it. As I said, all these membrane-bound organelles in eukaryotic cells are little tiny biochemistry labs. They sequester what they've got to do away from what else is going on in the cell because there's so much complex stuff going on that reactions over here might very well interfere with reactions over here unless they're separated from each other by membranes.
hence the nucleus, the mitochondria, the peroxisomes, the lysosomes, the glycosomes, and the list goes on, okay? Vacuoles, this is in plants, of course, the water vacuole that I showed you about, and I encourage you to get yourself an African violet, right? Yeah, okay. They are just basically a compartment of water. They provide the water needs for the cells in plants. They also, though, give the pressure to stretch, as I said, to stretch the leaves out. So anytime your house plant, doesn't matter whether it's African violet or not, anytime it gets all droopy, 9 times out of 10, now it may be sick, it may have a virus or millbugs or something like that. But 9 times out of 10, if it gets all droopy, it's because it needs a little bit of water, okay? And it'll absorb it and fill up that water vacuole, and the leaf will pop back up. All right, inside cells. As if stuffing the cell with endoplasmic reticulum weren't enough, sometimes I'm kind of floundered at how much stuff is stuffed in the cells, particularly eukaryotic cells. In animal cells, there are 3 categories of cytoskeletal elements. I can remember when these pictures came out, back when I was a grad student, we normally our stains look like black and white, or they might be red and blue, and they weren't fluorescent. And then all of a sudden, we started figuring out how to attach fluorescent dyes to things, and the journals started publishing these color pictures, and grad students would be looking at them in awe of how magnificent they were, but also how much they reveal. I mean, check out these things, these structural elements that are inside. These are all fibers, if you will, and we'll talk about them. So microfilaments are the simplest. They're the smallest, as the name implies, microfilaments. They are, sometimes they're simply referred to as actin filaments, although there are some microfilaments that are made of things other than actin. What's actin? You've heard of actin before. Where have you heard of actin? Who's an athlete in here? Your actin filaments have a great partnership with your myosin filaments doing what? Contracting your muscles, right? Yeah. And those are the athletes also that. But nevertheless, actin is important in terms of motility. It's also important just simply in terms of structure, and they tend to be, the actin filaments tend to just be on the inside. You can see them running right there on the inside of the cell membrane. So this is supposed to be the top and the bottom of the cell kind of cut away so you can see what's in it. They tend to be just underneath the surface. They're involved if you have cells that need to have any kind of motility, like some of the glandular cells. You don't want your salivary glands just squirting out digestive enzymes all the time, right? And so the openings where they would are often surrounded by a ring of actin and myosin, and they can squeeze down and keep that from happening. You understand? And there's other uses of them as well that you'll hear about if you go through courses going forward. They also are what provide the shape of any kind of cells that have a particular shape. This applies more to some of the protozoa, than say your liver cells. Your liver cells, their shape is pretty much dictated by the surrounding cells present in them, okay? But if you're talking about a paramecium or something like that, they have fairly elaborate external morphology, external shape, and it's the actin filaments that are under the surface that maintain that. They're also involved in the way, you understand what a amoeboid movement is? An amoeba, the way it moves, kind of a flowing movement. We figured this out in recent years. When I was in college, it was just a mystery. They talk about that, and we go, we don't know how this happened. They just flow along, okay? What happens is the actin filaments actually extend forward. They grow actually forward, and the amoeba attaches itself and then pulls you'll notice they're made of little monomers here, pulls the monomers off the back, and it's just done in a kind of a continuous fashion. That's the way that an amoeba moves, or a white blood cell for that matter. So those are the microfilaments, and you see the things that they're involved in. They are the flimsiest, if you will, of the cytoskeletal elements. But they can provide, these are protozoa, they can provide protozoa with rather elaborate morphologies, architectural structures on the outside of the body. Intermediate filaments. These are not the largest, but these are by far the strongest of the cytoskeletal elements. They basically run inside. You'll notice that they're attached to the cell membrane. They run throughout the cytoplasm of the cell. They're attached to the other cell membrane. They're attached to each other. They're actually what allow you guys to be sitting there on your chairs, padded though they are, and all of your butt tissue is not just liquefying. Oh, what do we have here? An educated wasp. That's cool. Learn Yeah, they provide the strength of your cells, and I'll show you a diagram that will give you kind of a best idea of that in just a minute. So they provide, strength and cell attachments. They're also involved in the attachment of one cell to another. Your cells would come apart if it weren't for intermediate filaments being involved. And then finally, microtubules are the largest, and they're kind of intermediate in strength. They are involved in motility. If you look inside the cilia or the flagella of a paramecium, then you'll see this arrangement. It's called 9+2. Yeah, I think I'll have a diagram of that coming up in just a minute. But they actually slide against each other, and that's what causes the flagellum or cilium to beat like that. Okay, so if I have a couple of microtubules and they're attached to each other by protein threads, if they move like that, it's going to cause the cilium to sway. So that's the way that works. There are also the spindle fibers that we see in mitosis and meiosis cell division. Those spindle fibers are microtubules, and microtubules are the way that things get around in cells. Neurotransmitters, you got these big old long nerves in your leg, okay? The cell body's up here at your hip, but you need to wiggle your toes, all right? You're gonna have neurotransmitters down there to do that. And so what happens is the neurotransmitters are made up near the nucleus, and then these little motor proteins get the big vesicles of those neurotransmitters and basically walk them down microtubules. It's kind of a cool process. You can see the 9+2 arrangement of the microtubules inside a cilium. So we got 9 pairs, 1, 2, 3, 4, 5, 6, 7, 8, 9, plus 2. You see where I'm with that? I might ask you that on a quiz at some point. So 9+2, let me see what it means. These are the linker proteins I was talking about. Here's a good example of what I was talking about earlier, the dynein and kinesin or motor proteins. They're actually some of the neatest proteins ever. They have little feet, for crying out loud, and they actually walk along the microtubules. So here's one carrying neurotransmitters down to your toes, so you can wiggle your toes or whatever. Associated with the microtubules are little items called centrioles, or actually centrosomes, the pair of them. You learned about centrioles in high school? I don't think you've heard of them before? Okay, so kind of during interface, sequestered over at the side of the cell is this little, oh crap. The other day there were like 17 markers. Where are they? So the centrioles are arranged like this. Remember in the cilia, they were doublets, they were pairs of microtubules. Here they're trios of microtubules, okay? But there are these two groups of microtubules, 9 of them, like so. They're arranged at, I'm not drawing all 9, but they're arranged at right angles to each other, basically. And they're made of tubulin. It's the same protein that the microtubules regularly are. They're involved in production of microtubules, except they themselves just sit there. They have a surrounding of a little area of little bits of tubulin, again, that protein that microtubules are made of, that are called nucleating sites. And anytime the centrosome, containing the 2 centrioles, wants to send out a microtubule, it actually shoots out from these nucleating sites, from these little rings on this. It's not a membrane, but they're kind of just arranged in space around the centrioles. And we call that structure, the whole structure, the centrosome. But it's these places that the microtubules shoot out from, send it down the nerve cell for a highway to your toes so you can ship neurotransmitters from the ends of the cell during cell division, so they can attach to chromosomes and cause the chromosomes to pull apart in mitosis. So, interesting. Okay, what in the world is going on here? This guy was kind of a famous person. I've looked his name up before. I don't have it on the tip of my tongue right now. Back in the 1800s, he had an interesting anatomical thing about him, obviously, okay? And it turns out that you have a lot of structures. So far we've been talking about structures inside cells, so organelles, cytoskeletal elements, and plasma reticulum, all that kind of stuff. That's not where it stops. There are lots of structures on the outside of cells as well. Structures that hold the cells together. structures that provide, you know, some stretch, but some stability to the skin, for instance. This guy, there is a condition, it's not a fatal condition or anything, but there are some people whose skin hangs much more loosely than other people's skins. I imagine when he got to an old age, he was probably kind of grotesque, okay? But, and as you get older, some of these connections between the collagen fibers do begin to loosen. So, you know, not too bad yet, but you probably, your ancient granny or something probably has a little jiggly, you know what I'm talking about? So, you're all looking at me like you don't have any idea what I'm saying, okay? You know how people get a little saggy when they get older? It's because the connections between the collagen fibers begin to kind of loosen a little bit. And some of you may have somewhat looser fibers than others. This guy was he was, I guess you could have said he was kind of a vaudeville, but he was also a political commentator and a comedian, okay? And he got his foot in the door by the fact that he could stretch his skin out to bizarre fashions like this, and so that was kind of an eye-catching thing. But then he would do that with, I think he started out kind of in the carnival routine, but as he started telling jokes and taking stabs at political figures and that kind of stuff, he became increasingly popular and eventually was touring Europe and performing before crowned heads of Europe and insulting them right there in their throne rooms and getting away with it because he was such a celebrity, if you would. But at any rate, what's happening there is that his collagen fibers just don't attach very well. He probably bruised pretty easily as well. Ages ago, when I was a teenager, there was this girl that she could stretch her skin and somewhat like that, and she said she really bruised easily, so she didn't like to do it. And I said, you bruise easily. Of course, I was a 17-year-old boy, and number one, I thought I knew everything, and number two, I didn't hardly know anything, and number three, she was a cute girl, okay? So I said, you know, she said, yeah, if I even bump into something, I'm likely to bruise. And so what did I do? We were sitting there at the swimming pool, so what did I do? I poked her a couple of times on the knee, and what do you suppose happened? There was a bruise there, and I'm going, okay, well, I'm shot down now. But at any rate, so there are conditions that involve the extracellular structures that produce some unusual results. If you were to look under his skin, you would see these cells that are called fibroblasts. They're the ones that produce the collagen fibers, and you can see that a lot of these collagen fibers are secured to each other, right? You can sense that. In his face, under his skin, they would be much less so, okay? Plants have rather profound extracellular structures. Plants don't really have the same kind of cytoskeletal elements that I was just talking about. They do have microtubules that, you know, during mitosis and all, but they don't really have so much in terms of actin filaments, microfilaments, and their intermediate filaments are limited as well. But outside the cells, they have cell walls made of cellulose and other structures. And so you can kind of see, even between all of these cells, you see the cellulose cell wall. Here it's been stained with some fluorescent dyes. There are some tissues in our bodies that produce a great deal of extracellular matrix, extracellular substance. Our bones are mostly extracellular. We do have cells inside our bones called osteocytes. You ever heard that term, osteocyte? The prefix osteo means bone. So an osteologist is someone that who studies the skeletal, repairs skeletal things. So anyway, this is basically cartilage right here, which is related to bone. All of this stuff is collagen, the matrix, and all of the cells are in little tiny holes called lacunae. Lacuna is the singular, and so these are the cartilage cells inside there. Same thing with bone; they look somewhat similar, also a little bit more arranged. All right, let's move on to the next topic. We're doing fine here. We've got basically, in case you hadn't been looking at your calendar, what happened a week from today? Yeah. And so I've got just the right amount of today's and the next class period. So we'll talk a little bit about cell membranes. Here's the roadmap. Back when I was in high school, the first real good model of the way the cell membrane is put together was established by a group, the Sanger group it's called. And anybody know the name of the model? It's still in use, of the cell membrane. Have you ever heard of the fluid mosaic model? Remember, the cell membrane is this phospholipid bilayer, and the only thing that holds that phospholipid bilayer together is the fact that it's made of phospholipids, which have hydrophobicity and hydrophilicity, right? And I showed you how they sort themselves. The result of that is that, yes, it does make a membrane, but it's a very fluid membrane, and it has proteins of a variety of sorts, we've come to understand, that float around with it in it. There, in the original article of the fluid mosaic model, Sanger and his colleagues said that they likened it kind of poetically unto protein ships afloat in the liquid sea, was the way they put it. In other words, they can move around. So we'll look at a little bit about the way membranes are put together. Skip one there, there we go. Okay. So here's a cell membrane of one cell. Here's another cell over here with its cell membrane. So what do we see? We see a variety of structures. First of all, here's the phospholipid bilayer. Okay. All these little things that look like little mice, those are cholesterol molecules. We tend to think of cholesterol as something you need to avoid, and it isn't true, but your body makes cholesterol, and the cholesterol that's embedded in the cell membranes basically provide them with some of their plasticity. What happens to lipids? Your cell membrane's a lipid, right? Phospholipid. But what happens to lipids when they get cold? So think about the butter in the butter dish. If it gets real cold, what happens? It gets real hard, doesn't it? What if the butter gets kind of warm? It can melt, right? You don't want your cell membranes doing either of those things, do you? The cholesterol that's embedded in the membrane actually stabilizes that, okay? When you take cell biology your junior year, if you take cell biology, Dr. Conan will have a lot more to say about that, certainly. It's an important part of the cell membrane structure. Other things are various types of proteins that extend through. You can see this one and this one have little, that's supposed to, cartoonishly indicate that they're moving along, like a runner or whatever, with speed marks behind it. So they can move around in this membrane. Some of them are called integral proteins, meaning they go all the way through the membrane. Some of them are external, and some of them are internal, attached to one or the other side of the cell membrane. Some of them are big constructions like this, which are actually a channel, which would allow certain things to get into or out of the cell. Some of them have, often the receptors for various substances in cells are carbohydrates. So this would be a carbohydrate attached to a protein molecule, okay? And this would probably enable it to recognize something, depending on what. So you can see here's some that are partially integral, just the inner side. That just shows you, again, the phospholipid arrangement with the aqueous environment, the water environment out here, and the water environment here. Integral membrane proteins are partially or totally inserted in the phospholipid bilayer. Peripheral ones tend to be attached to the inside or the outside. I already mentioned carbohydrates are often involved in recognition signals between cells. And then one other type, there's a variety of ways that cells stick together. One of the simplest ways is integral proteins, like this one and this one, that then attach to each other in the space between those cells, okay? And you can see how that would hold cells together. There are all kinds of cell-to-cell attachments, as we're going to see. This is just basically one of the simplest ones. So let's talk about these. There's three categories. There's some subcategories between these, but there's three basic categories of cell membrane attachments, if you would. One is called a pipe junction, okay? This is not that strong of a connection, but you'll notice, like this is the cell membrane of one cell, and this is cutaway. This cell would extend all the way over that way, and then this other membrane is just a section out of the cell that would extend all this other way. So you see there are two cells held together like that, okay? And they're stitched, it almost looked like someone took a sewing machine and stitched them together, didn't it? Or I could take a couple of pieces of paper, these quizzes, for instance, and stitch them together with a sewing machine, and it would be hard to insert another piece of paper between them. At the same time, it'd be real easy to rip them apart, wouldn't it? Because they're made of paper, and that's the same with these. What these tight junctions do is they prevent things from seeping down between the cells. Okay, now why is that important? Well, if you look in your GI tract, for instance, your intestine, these are among the main cell-to-cell connections in the intestinal lining. How much do you not want the contents of your intestine seeping out from the inside of your intestine?
you probably want that a lot, right? So, you'll find lots of tight junctions in that sort of a circumstance, so that the contents of one structure or one cavity, not just intestinal cavity, it's in other places as well, so the contents of one cavity can't seep between the cells to the next cavity. So they're about water tightness, if you will. Any questions about those? Desmosomes are all about strength, okay? So desmosomes are ones that attach themselves indirectly to intermediate filaments. Keratin fibers here would be a type of intermediate filament. So check it out. Here we have keratin fibers that run from this side of the cell all the way over to the other side of that cell, okay? And then you have the cytoplasmic plaque, as it's called. It's a protein plaque. And then there are these connecting fibers that run through between these. And these are the ones that give strength to your cell-to-cell connections, okay? These are not about keeping things from leaking between the cells. These are keeping the cells from popping apart. You know, I'm a pretty big old guy. If I sit up here on this table like so, there's a considerable amount of pressure being applied to my rear end tissues, right? And if I didn't have these desmosomes, this type of structure, pretty much the cells in my butt would be mush, okay? So, you follow the idea? That's what keeps the integrity of your cells together here strongly. We're going to see that there is another manifestation of desmosomes called hemidesmosomes, and I'll show you a picture of what they do in just a second. And then finally, gap junctions actually allow stuff to go between cells. You see, they look almost like little flowers, but they create a little tube, don't they? A hydrophilic channel. Again, what's that word hydrophilic mean? It likes water, and anything that's dissolved in the water will be able to pass from one cell into the adjacent cell. This is very important in a variety of circumstances. You remember those, I was showing you those in cartilage, those cells that were inside the lacunae but separated by all the matrix? They have to be able to communicate with each other, and they have these long tendrils that reach out and actually touch, just like my fingers are now. And at those places where they touch, there's tons of these gap junctions, okay? Same, even more so in bone cells, okay, to allow things to pass. Your heart muscles have a lot of these gap junctions. You may be aware that your heart doesn't really have that much nervous tissue. I don't know how much you study. You'll get into this more in A&P next semester or next year. But your heart really doesn't have a whole lot of innervation, nervous system. What it does is the heart muscles, the cardiac muscle cells, are actually able to conduct electrical impulses. And the reason is, I mean, you know that electricity can pass through water, right? Okay. The reason is there are these places between the cells that are just chock full of gap junctions. And so if there's an electrical impulse here, it's going to flow through those channels into the next cell, and the next cell, and the next cell. And so what happens is, you probably heard of pacemakers take the place. If you have there are a couple of nodes in your heart that start a nervous impulse, okay? The vagus nerve comes down and stimulates one of them, and what it does is it sends out an electrical impulse into the cardiac muscle cells, and then that cardiac muscle conducts the nervous impulse all the way down through the chambers of the heart, okay? And so it's because of the ability of these gap junctions to allow that electrical impulse conduction. Pretty cool process. When you take A&P, it's going to get even cooler because they do it in a very synchronized fashion. Everything's working right. Yep.
Does only the heart have gap junctions?
Well, no, I just told you there's other the connections between some of the cells like osteocytes in the bone and chondrocytes in the cartilage. There's lots of other instances where the gap junctions certainly do occur. And I think we'll probably see some even in the interconnections between some of the intestinal cells. I'm going to show you in a minute. They're fairly common, but there are some tissues that really make big time use of them, if you will, is the point of the cardiac muscle. All right, so here we have a cutaway of your GI tract. Okay, this is a small intestine. I can tell because it's got microvilli. The large intestine doesn't have the microvilli. But in any case, what all do we see here? So we see the tight junctions right up here at the top, don't we? Where's all the digested food product that's in here, the lumen? You all understand the word lumen? Again, into that toolbox, lumen means a cavity, a space or something. It can be the lumen of a blood vessel, the lumen of your stomach, inside something. So here's the lumen where the food is, and that you don't want seeping down between these cells. Here's the tight junctions to keep that from happening. Here are the desmosomes and the intermediate filaments connecting. Can you see how they are going to, if you compress these cells, they would compress up to a certain point, but eventually these intermediate filaments would just stretch out to their full length and stop the stretch, wouldn't they? They maintain the integrity of the cell. And then, here are the gap junctions that we just, mentioned here that allow passage of nutrients, cytoplasm between these cells. And then the so-called hemidesmosomes are, you can see they're like, well, hemi means half, and so you can see that they're attached to the, basically to the basal membrane here, and they hold these cells in place. These are intermediate filaments, and they go up and attach to these desmosomes up here to hold everything securely. Any questions about that? So there's the tight junctions, the desmosomes, and then the gap junctions. A little bit about the way that stuff moves across membranes. You know, if I can get out a little early today, this is the slide I wanted to get to for sure. So, cell membranes are all about keeping stuff out that needs to be kept out, okay, and providing the integrity of the cell, but also keeping stuff in that needs to be kept in. But what else do they need to do? They need to allow things in, nutrients and things at certain times, oxygen, whatever, okay? And they also need to get stuff out, waste products or possibly enzymes that are being secreted to the outside. So there are three general categories of the way things get into and out of cells. I'm going to explain these now, and it looks like you will get out a little bit early today. I'm impressed with myself for getting to what had to be my stopping point. One is simple diffusion. What happens here is whatever you're talking about moves with the concentration gradient. Now, what do I mean by that? We got a membrane, okay? You're assuming that something's moving across a membrane. Do a diagram. So here's a cell membrane, and you've got some kind of molecules, let's say oxygen molecules, for instance. And the oxygen concentration over here is 5%, and the oxygen concentration over here is 2%, okay? With the concentration, oxygen can pretty much get through a cell membrane pretty easily, okay? So it's going to simply diffuse in the direction from higher to lower concentrations. Does that make sense? So that is with the gradient. Against the gradient will be the other way. That's a lot harder, but we have mechanisms for taking care of that. So in simple diffusion, there are not that many things that can simply diffuse. Water can, oxygen, some other elemental type of materials can diffuse across cell membranes. They go with the concentration gradient. They don't require any kind of special membrane protein, a carrier protein or a transport molecule or something like that. And it's not specific. If they can get through the membrane, they will go for an area of higher to lower concentration. You with me? They don't require any additional energy other than that concentration gradient. The second category is also with the concentration gradient, but these are things that you don't necessarily always want to let into the cell just, uniformly in mass, okay? And there typically are membrane proteins involved with this. So if there's a lot of sugar on the outside of a cell, glucose let's say, it may or may not be allowed into the cell. If the cell needs glucose, then those glucose molecules will attach to very specific membrane proteins. Specific meaning there are membrane proteins for glucose, and there are membrane proteins for trioses, and there are membrane proteins for practically separate ones for amino acids and things, practically anything that needs to get inside a cell. And they'll go just like this from an area of higher to an area of lower concentration, but those membrane proteins are very specific, those carrier molecules. And on Tuesday, we'll be talking somewhat more about those specific molecules. So again, no extra energy required other than the concentration gradient. Now, sometimes you need to get stuff into or out of a cell, okay? If a cell is filling up with metabolic waste products, they're going to accumulate, right? Okay? And they're going to have to be shipped out. That's fairly easy. Possibly some of them can be done with facilitated diffusion. But in some cases, they may have to be transported out. Moreover, if the bacterium or the cell that's doing this is an organism that lives in, say, a sewage treatment plant, where's the more waste product? On the inside of the cell or the outside of the cell? On the outside of the cell, okay? So it's going to have to get rid of its own waste products against the gradient. Does that make sense? And so that requires ATP. We'll look at some things where that occurs. I'll give you some examples on Tuesday for that. Similarly, if a cell finds itself in an environment where there's fairly low amounts of, for instance, glucose, but it needs all the glucose it can get, it may have slightly higher glucose concentration inside the cell, but it's still gonna get those glucose molecules, okay? Because they're resources that it may need down the road. But what it's gotta do is it's gotta pump it against the gradient, doesn't it? So again, it's gonna require ATP. And these, once again, these membrane proteins are extremely specific. I mean, the one that carries, the amino acid tryptophan will not work with leucine, you see what I'm saying? So there's thousands of these membrane receptors that allow that kind of movement. So, on Tuesday, we will talk about these aspects and what they mean in relation to osmosis and osmotic pressure. And then we'll finish that up, and then we'll also talk a little bit about energy considerations on Tuesday, and then exam on. You want to know about the exam? Okay, I think I've told you this before, but just to remind you, it will be, I do my exams, when I do my exams, I do not have the book open beside me, okay? If I have to open it to find out exactly how something might have worded, but I don't normally do that. I basically have my PowerPoints available and my understanding of what we talked about in class available, obviously. And the questions will be drawn from rather substantial test banks that I have, which I add to every time. I'll add some new questions, and they will be, the test will be multiple choice questions, 50 multiple choice. And I think I told you for the first day of class that I do item analysis, so that if more than half the class misses any given question, then it becomes a candidate for getting tossed out. Usually it'll get tossed out, but usually I can look at it and go, well, boy, I worded that bad. Or, I remember teaching that to them, they'll never understand this aspect of biology, so I just give them the point back. But sometimes I look at it and go, yeah, no. I'm trying to think of a good example right now, but I mean, how many people missed the question about the pH 4? Okay, so that's one, not more than half the class missed it, but more than half the class has missed it, then I'd go, I don't do item analysis on imagine, but on the test, if more than half the class misses that one, I go, yeah, no, I think I know that, sorry. But 9 times out of 10, I'll take the ones that are missed by half the class, more than half the class, excuse me, and ditch them. And it adjusts the scores appropriately. I can almost assure you that the average on the first exam will be somewhere in the mid-high 70s, which is tough. There'll be plenty of A's, there'll be some people that'll be disturbed, but we'll talk about that after the okay? And tutoring, if you feel like you need it, the night before the exam is not the best time, but now is beginning to get late in the game, but if you were to go to the CASA or the CAE, then, or they have, well, it's a race, but they have, actually, I think it was last night, wasn't it? Yeah. They do have drop-in tutoring as well, but if you need a tutor, don't be bashful, get a tutor. Your tuition already paid for it, so for crying out loud, make use of it. Okay? Go on then. And have a good weekend. Have a good weekend with the understanding that you have tests coming up next week. Probably not just in biology, right? Pretty much, unless I decide not to. And I won't ever have one today, the class after a test. So we'll have a test on Thursday, I won't give you a quiz on Tuesday. And each quiz will always be since the last quiz or the last quiz. One of those famous forms, boy,